Okay, so continuing respiration. I want you to think, when you hear the word respiration, think electron transport chain. Because remember, that's what respiration is. Respiration has nothing to do with oxygen. In humans, yes, we respire oxygen, but at the most basic biological sense, respiration means that an electron transport chain is in use. And so here is a diagram of an electron transport chain. This would be in the membrane of the mitochondria. Okay, this is in one of those internal membranes in the cristae of the mitochondria. On a prokaryote doing respiration, this would be on their cell membrane. But in a eukaryote doing respiration, it's on the membranes of the cristae of the mitochondria. So there are these 4 proteins that are integral membrane proteins embedded in the membrane. Then there is a carrier molecule called quinone, shown in orange with a little Q on it. And then there's another protein that is membrane associated, although not membrane embedded. It's membrane sort of soluble. It moves around in there. And that's cytochrome C. Okay. And these proteins are called complex 1, complex 2, complex 3, and complex 4. And then we have quinone and cytochrome C. And so what respiration is doing is it's taking the electrons off of NADH and passing them onto another molecule. Okay. This happens to be an aerobic or oxygen-based respiration electron transport chain, but there are anaerobic electron transport chains. So what happens in electron transport? NADH arrives at complex 1. So remember, there was the orange part is the cristae, or sorry, the orange part is the matrix of the mitochondria. The blue on the top is the inside the cristae in the intermembrane space. And so this is embedded in the membrane of the cristae. So we've had the citric acid cycle, TCA, go on in the matrix of the mitochondria and produce all this NADH. We can also ship NADH from the cytoplasm into the matrix of the mitochondria that was produced through glycolysis. Okay. That NADH arrives at complex 1 and it gives up those 2 electrons that it's carrying to complex 1. We have now restored NAD+ from that molecule. Those electrons, as they pass to complex 1, because they are high energy, they allow complex 1 to pump some protons from the matrix into the intermembrane space. And so you're seeing those protons moving as this black arrow. We're concentrating hydrogen ions in the intermembrane space. The electrons are then passed from complex 1 to the molecule quinone. When quinone gets reduced by those electrons, it then picks up a hydrogen and it carries those electrons and that hydrogen to complex 3. It completely skips complex 2. Okay, it goes straight to complex 3. Quinone becomes oxidized and donates electrons to complex 3, which becomes reduced. And it also gives up that hydrogen it was carrying, and it releases that hydrogen onto the intermembrane space. Then the electrons go from complex 3 to a molecule called cytochrome C. That protein, cytochrome C, is reduced by the electrons from complex 3. Cytochrome 3 gets reduced, carries the electrons to complex 4, where it is then oxidized, and reduces complex 4. So cytochrome C gets reduced by complex 3, carries the electrons to complex 4, where it gets oxidized. That then reduces complex 4. When complex 4 is reduced, that allows it to pump some more hydrogens from the matrix into the intermembrane space. And complex 4 finally donates those 2 electrons, which have given up all their energy and allowed the pumping of lots of hydrogen ions. It donates those 2 electrons onto an oxygen molecule. The oxygen molecule then combines with some hydrogen to make water. So in the process here, we've taken electrons from each of the NADH that we've produced through either glycolysis, acetyl-CoA pathway, or TCA. We've donated all those electrons to complex 1, which is then passed them down this electron transport chain. In the process of that, the energy from those electrons has allowed the pumping of hydrogens across the membrane to create a proton modem force. This is a electrochemical gradient of hydrogen ions across the membrane. Those electrons then are finally dumped onto an external electron acceptor, in this case, oxygen. Oxygen is one of those molecules that is very easy to reduce, has a very low resting potential, and so it can accept electrons from a lot of things. And so it makes a great acceptor at the end of this electron transport chain. Remember, in the TCA cycle, we also made FADH2. FADH2 can donate its electrons to the electron transport chain as well. FADH2, however, donates its electrons to complex 2, which complex 2 then passes them to quinone, and then the pathway is the same. Quinone to complex 3, to cytochrome C to complex 4, reduces oxygen to produce water. Okay. In both ways, we restore oxidized forms of NAD+ and FAD. So they're ready to be reduced again, and we can continue on making ATP through glycolysis and TCA. Okay. So that is an electron transport chain, and that is an aerobic electron transport chain simply because it uses oxygen as a terminal acceptor. Each time the electron gets passed through this electron transport chain from one molecule to the next, it drops in energy. So here we have on the y-axis the free energy change, so the delta G relative to oxygen, to O2, in kilocalories per mole. So electrons from NADH are very, very high energy. They have a very high delta G, very positive. And so they pass to complex 1, then to quinone, to complex 3, to complex cytochrome C, to complex 4, and from complex 4 finally on to O2, which has a very, very low resting potential. Okay, so this is each passage of electrons here is a series of redox reactions. Electrons from FADH2 begin with a slightly lower energy level, so they can't be passed to complex 1. So instead they're passed to complex 2, which can then donate them to quinone, to complex 3, to cytochrome C, to complex 4. Okay. So each time the electrons get passed, they step down in energy level. And that energy, where it goes, it doesn't disappear. So energy goes to moving hydrogen ions across the membrane to create this proton motive force. Now, this proton motive force turns out to be really important for overall energy production in the cell. So there's a hypothesis called the Mitchell hypothesis, or the chemiosmosis hypothesis, after the scientist who came up with it. And Dr. Mitchell, she had this very simple hypothesis. She was looking at the electron transport chains and ATP production. And she was asking, how are the electron transport chain and ATP production linked? So her hypothesis was that the linkage is indirect. The electron transport chain creates a proton motive force that drives ATP synthesis through an ATP synthase. The null, or the alternative hypothesis, is that the linkage is direct. By running this electron transport chain, there is an enzyme there that directly performs substrate level phosphorylation of ADP to produce ATP. And the way she tested this is she used something called bacteriorhodopsin. It's a protein from archaea that pumps protons across the membrane when struck with light. And so what she did is she produced these vesicles, which, remember, vesicles are just bubbles of lipids, phospholipids. She produced these from artificial membranes. These vesicles contained ATP synthase in them, and she added bacteriorhodopsin to these vesicles as well. Then it's simply a matter of exposing these vesicles to light. And so when light hits bacteriorhodopsin, it causes the pumping of hydrogen ions out of the vesicle across the membrane. And then if those hydrogen ions can flow back through the ATP synthase, then ATP will be made. So the prediction of the chemiosmotic hypothesis here is that ATP will be produced within the vesicle. If the alternative hypothesis is true, then she will lack the protein necessary for that substrate level phosphorylation, and no ATP will be produced. And the results were that there was a lot of ATP produced within the vesicle, even without a full electron transport chain there. So the electron transport chain is not doing substrate level phosphorylation. It is creating a proton motive force. Okay, so it's an indirect linkage. So this indirect linkage is this protein called ATP synthase, also known as the F1F0 ATP synthase. It is a protein embedded in the membrane of the matrix of the mitochondria. And what it does is it allows hydrogen ions to flow down their gradient. So the electron transport chain created this concentration of hydrogen ions in the intermembrane space. The ATP synthase allows them to flow back through the mitochondrial membrane into the matrix. And as it does that, they're going down their gradient, so they have energy. And they provide that energy to turn this ATP synthase. And as this ATP synthase turns, it combines an ADP and an inorganic phosphate together, and it uses the energy of those hydrogen ions moving to make ATP. Okay. So it's basically a little rotary motor, and every time this ATP synthase turns, every time a hydrogen ion flows through, it turns a certain amount, and that causes the reaction of ADP plus phosphate to make ATP. You can make from the 10 NADH and 2 fat H2 produced through the oxidation of a single glucose, from glucose to pyruvate to acetyl-CoA through the citric acid cycle. So take those 10 those 10 NADH and your 2 fat H2. If you pass them through electron transport and use oxygen as your acceptor, you can make an additional 25 ATP using this ATP synthase. So from a single glucose, here's how much energy we can get. Using aerobic cellular respiration, we can break glucose down into 2 pyruvate, get 2 ATP and 2 NADH. We move pyruvate into the matrix. We convert pyruvate into acetyl-CoA and produce 2 NADH. We take that acetyl-CoA through the TCA, produce an additional 2 ATP, 6 NADH, and 2 fat H2. We take all of this NADH and fat H2 and pass it through our electron transport chain to produce a proton motive force, consume that proton motive force through the ATP synthase, and make an additional 25 ATP. So your mitochondria can make a total of 29 ATP per molecule of glucose that they break down using respiration. That's a pretty amazing yield of energy. Now, this is aerobic respiration because this is respiration that happens in the mitochondria of humans. Many organisms, especially prokaryotes, do not have to breathe oxygen. They are anaerobic, but they can still respire. And so here's a simple example of anaerobic respiration. This can happen in E. coli. So E. coli can breathe oxygen if it's available, but you can also put E. coli in an oxygen-free environment, and it can still respire. What it will do is it will break down glucose through glycolysis and the TCA cycle to produce a bunch of NADH. And then it will take that NADH and will pass them to this alternative electron transport chain. So this is on the membrane of the cells. So here's the inside of the cell at the bottom and the outside of the cell at the top. So it's going to take NADH, and it's going to donate electrons from NADH to this FES complex. That's going to pump 4 hydrogens out of the cell. Passes the electrons to a quinone. Quinone will carry an additional 4 hydrogens out of the cell. That quinone passes electrons to something called cytochrome B, which then passes them to something called nitrate reductase. Nitrate reductase is going to take those electrons and dump them onto nitrate to make nitrite. Nitrite is a toxic byproduct, and that gets pumped out of the cell. As a result, the cell has produced a proton motive force that it can then consume through an ATP synthase to make ATP, all in the absence of oxygen. The big difference is this terminal protein, this final protein in the electron transport chain. Does it put those electrons on oxygen or on something else? In this case, it is nitrate. Right? So that's an anaerobic respiration. There are lots more. There are things that breathe sulfur. There are things that breathe nitrate to nitrite to NO gas to nitric oxide, all the way to N2 gas. And so there are things that breathe methane. There are all kinds of alternative respirations among the prokaryotes. I just want to point out one to remind you that respiration is not always aerobic. Okay, so that's respiration. We make a bunch of NADH and fat H2. We pass those high energy electrons to an electron transport chain to reduce an external molecule, such as oxygen, nitrate, sulfate, etc., and produce a proton motive force. That restores our pool of NAD+, so that we can continue glycolysis. The molecule in respiration is called external because we have not previously used oxygen, nitrate, sulfate, or whatever in the glycolytic pathway. Okay. The other option is, instead of respiring, is to ferment. So in fermentation, we're going to take NADH and fat H2. We're going to pass those high energy electrons either to pyruvate or another molecule derived from it, and that's going to restore the NAD+ pool. So example of how this works. In humans, in our muscle cells, if you do aerobic exercise, for example, you're running or riding a bike for a long time, to the point that you're short of breath, right? What you're essentially doing there is you're starving your muscle cells for oxygen. You are using it up faster than you can transport it to your cells. But the cells still need energy, so they're still going to run glycolysis. So what they do is they break down glucose into pyruvate through glycolysis. You produce two ATP and two NADH. Now, if you use up all your NAD plus and make it into NADH, the cell dies, can no longer make energy. So to keep that from happening in the absence of oxygen, because we cannot breathe nitrate, what happens is they take pyruvate and they take the electrons off of NADH and they dump it back onto the pyruvate, and you make this molecule called lactate or lactic acid. That restores NAD plus and allows the cell to continue through glycolysis and make this small amount of ATP in the absence of oxygen. So it does not produce any additional energy, but it keeps the cells alive, right? You don't get any energy after pyruvate. It's essentially a loss. You're burning the energy of NADH. But what you're doing is you're allowing glycolysis to continue to make the ATP of glycolysis. Other cells can ferment as well. E. coli can ferment. Yeast can ferment. Yeast ferment a slightly different way. They take glucose and produce pyruvate through glycolysis. Then they convert pyruvate into something called acetaldehyde, and they release carbon dioxide as a result. Then they take the electrons off of NADH and put it on the acetaldehyde, and they make ethanol. This is essentially the fermentation that's used to brew alcohol, brew beer, and other spirits. Okay, we take some sort of sweetened liquid, something that contains sugar, so grape juice to make wine, or other sort of corn, carbohydrate, mash, whatever. Allow them to use the glucose in that to make pyruvate, and then continue to ferment to produce ethanol. Bacteria can ferment to produce alcohol. Bacteria can also ferment to produce lactic acid. And that is what helps produce things like cheese, yogurt, bread, sauerkraut, kimchi, miso, soy sauce. All of these foods are the result of fermentation. And it's a result of bacterial or yeast fermentation in the food product. So these byproducts like lactate and ethanol, we happen to find quite yummy as humans. And so we use these organisms, yeast and bacteria, to ferment various foods to make additional food products. Okay. It's also often a form of preservation. Okay, so that is cellular respiration, okay, and fermentation. So if we look back at cell respiration, assuming we're looking at human cells, we have glucose broken down to pyruvate to acetyl-CoA through the TCA to produce a lot of CO2. Consequently, the carbon dioxide you breathe out when you breathe, that you exhale, is from your food. Think about that. The solid food you ate got broken down in your stomach into individual small molecules of glucose or starches or whatever, sent to your cells. Your cells broke that down into carbon dioxide, shipped that carbon dioxide back through your blood to your lungs, where you then breathe it out. I find that amazing. We are essentially breathing out the food we ate, the byproduct of our metabolism. It always amazes me. But what you have here is you have glucose. Catabolism is at the top. You have the food that you eat. Carbohydrates would go in through glycolysis to be broken down to pyruvate, acetyl-CoA, then through a citric acid cycle. What if you're not eating carbohydrates? What if you're eating fats and phospholipids, right? So you're eating an avocado, or you're eating something with olive oil in it. You're eating a fat. You break the fatty acid, the phospholipid, down into two parts, into the glycerol part and the phosphate part. The glycerol should sound familiar. You can convert glycerol into glyceraldehyde 3-phosphate. And you can ship that through part of glycolysis, not the whole thing, but at least part of it to make pyruvate, and then break it down through acetyl-CoA and TCA. You can take the fatty acids and you can break off 2 carbons at a time from fatty acids to make acetyl groups and attach them to acetyl-CoA and break them down through the TCA cycle to make some NADH to get energy that way. If you're eating proteins, you take amino acids and you break off the amine group, you're essentially left with these carbon compounds that can either be fed into, depending on how big they are, into produce pyruvate, to produce acetyl-CoA, or to produce some intermediate in the citric acid cycle. And you can extract some energy from that. If, however, you want to build things in the cell, you want to do anabolism, you can take any of the breakdown products that are along this metabolic pathway, and you can stop metabolizing them, stop breaking them down, and use them for other things. So glucose, before you put the 2-phosphates on it, you can make into glycogen or starch for storage. You can also take off pieces of the glycolytic pathway. You can make some of the precursors for DNA or RNA. You can make some of those ribose molecules you need. You can take acetyl-CoA, and you can use it to make phospholipids and fats to put into your membranes. And you can take things from the citric acid cycle, and you can use them to synthesize amino acids for protein synthesis. So cell respiration is central to both the catabolic and anabolic reactions of the cell. Things can be broken down to harvest energy, but things also can be funneled off of the pathway to build up cell mass and cell molecules. This is a simplified diagram. I like this one because it is a more true diagram of how this works, where the thick gray line in the middle is glycolysis, and then the purple is pyruvate, the orange is acetyl-CoA, and then you have the TCA cycle there. And then all these little dots going off are representing the different molecules in different parts of the pathway that you can ship off to and make things from. Okay, so lots of branch points and modularity in our metabolism. So an energy summary for glycolysis anaerobic respiration. We're going to take glucose, C6H12O6, plus 6 oxygen, and we're going to break it down to produce 6 carbon dioxide, 6 water, and energy. And the energy is going to come in the form of 2 NADH and 2 ATP from glycolysis, 2 NADH from pyruvate processing, 6 NADH and 2 fat H2 plus 2 ATP from the TCA cycle. And then, assuming that we're respiring, we're going to take all of those NADH and fat H2 and convert them into 25 more ATP from the aerobic electron transport chain. For a net total gain from a single glucose, you're going to get 29 ATP molecules out in your mitochondria. All right, that's it for respiration and fermentation.